Hi dear students, a warm good morning to all of you and welcome back to one more session of immunology. Today we are going to continue with the organs of the immune system, the part 2. In the previous session, we had mentioned about the primary as well as the secondary lymphoid organs, and we did come across about the bone marrow as well as the thymus, where the B lymphocytes and the T lymphocytes are being matured respectively. And we also came across the secondary lymphoid organs, the locations where the B lymphocytes and the T lymphocytes get exposed to the antigen. And among them, we do have well-developed systems such as the spleen, or the well-developed organs such as the spleen and lymph nodes and all that. But at the same time, we also do have certain lymphoid tissues which do play a role as the lymphoid organs. And today we're going to talk about the lymphoid tissues which are being involved in the secondary lymphoid organ as secondary lymphoid organs. Now, what do you mean by lymphoid tissues? The lymphoid tissues are some less organized tissues which are being found at different parts of the body, and they do play an important role in the immunological system. If you go to consider the lymphoid tissue, they are being found to be highly organized and loosely associated and cellular complexes, and sometimes they can be associated with specific locations or sometimes at different other locations. Now, for example, sometimes it can be associated with the skin and we call it as a skin associated lymphoid tissue, and sometimes it will be associated with the mucous membranes of different parts of our body, and then it is called the mucosal associated lymphoid tissue. And salt as well as malt, as it is being abbreviated, they are good examples of highly organized lymphoid tissues, and you can see that in this regions of skin associated lymphoid tissue as well as in the mucosal associated lymphoid tissue, there are specific areas where B and the T lymphocytes and sometimes dendritic cells are being present, and surrounding these areas, they are also being accompanied with the presence of antigen presenting cells such as the macrophages also. So, the lymphoid tissue is not exclusively for one type of immunity, that is either the humoral immunity or the cell mediated immunity can be the factors of humoral as well as the cell mediated immunity could contribute towards the factors in the lymphoid tissue. And sometimes you can also find certain loosely associated bronchial associated lymphoid tissues can also be present, where such cellular partitioning is not being present. So the primary role of these lymphoid tissue is to organize the WBC of the leukocytes and to increase the interaction between the innate and the adaptive arms of the immune response. Now, if you go to consider the macrophages, they come under the innate immune system, and the T lymphocytes and the B lymphocytes come under the what? The acquired or the adaptive immune system. So, a bridging up or the increasing of the interaction of the innate and the immune system is being brought about by the lymphoid tissue, because it contains both the members of the innate immune system as well as the members of the adaptive immune system also. So, the lymphoid tissue is an interface between the innate resistance mechanism and the adaptive immune system of a particular host. Now, let's go into the details of all these types of tissues. The first one which we are going to talk about is the mucosal associated lymphoid tissue. If you go to consider malt, it has been found in various body sites. Sometimes it might be found in the small intestine, and there it's called the Peyer's patches, or it might be found within your tonsils, in your appendix, or sometimes you can find it in the lamina propria of the intestines, or it can also be found in the upper airways, bronchi, or in the genital tract. So, where all these mucosal membranes are present, in most of the cases associated with these mucosal membranes, there are lymphoid tissues also. And based upon where they are being located, malt can be named by different names. Now, when it has been associated with your nose or the nasal tract, it is called the nasal associated lymphoid tissue. And which has been associated with your gut, it is called the gut associated lymphoid tissue. And if it has been associated with your bronchi, you called it as a bronchus associated lymphoid tissue. And sometimes it will be associated with the genitourinary tract also. And the main role of these mucosal associated lymphoid tissues is that both antibody producing cells as well as the T lymphocytes will also be present, and at the same time various antigen presenting cells will also be present over here. And but when you compare the malt with the other plasma cells in the spleen, lymph nodes, or in the bone marrow, you can see that the number of antibody producing plasma cells will be higher in the case of malt, in the regions of malt, compared to the regions in the spleen, lymph nodes, or in the bone marrows combined. So, what happens? In other words, you can see that the military of antibody producing plasma cells has been found to be very much higher, or the antibody production is being found to be higher in the case of the mucosa associated lymphoid tissues compared to the spleen, lymph nodes, and the bone marrows together. So how does they work? That's what we are going to talk about. If you're going to consider the mucosa associated immunity, uh, especially when you consider the mucosal membranes, usually there will be, now this is a mucosal membrane, and there will be epithelial cells which are being found, which would have like finger-like projections, microvilli-like, or any type of projections would be there in the mucous membrane. And at the same time, in between of these epithelial cells, there would be other cells called the M cells also. Okay, now what is the function of these M cell? It is the M cell, okay, which will endocytose or take in the pathogen, and it will release it into a particular pocket which contains the helper T cells, B cells, and the macrophages. So if you consider a mucous membrane, in between that we have the M cells. And these M cells on these mucosal membranes will take in the pathogen or endocytose the pathogen inside. And it will enter into a pocket where you have the T helper cell, B cell, as well as the antigen presenting cells. And within these pocket, what happens? The pathogens will get, now the antigen presenting, the macrophage is an antigen presenting, it is an antigen presenting cell. It will phagocytose it and try to destroy it. And at the same time, what happens? These antigens are also being presented to the B lymphocytes, and the T helper cells also will interact with the B lymphocytes also. So a combined action of these three types of cells will be carried out within the M cell associated pocket. And what happens? The antigen is getting transported by the M cell to the organized lymphoid follicle. This is the follicle what we consider here. Okay, and the lymphoid follicle will contain B cells as well as what happens, and when the B lymphocytes get activated, they start producing what? Plasma cells. And we know that the plasma cells are the ones which are responsible for the formation of immunoglobulins. And the plasma cells will start producing immunoglobulins. And in the case of mucosal membranes, you can see that the type of antibody which has been formed here are the secretory immunoglobulins. And so what happens? IgA type of immunoglobulins will be formed. In the case of immunoglobulins itself, there are different types of immunoglobulins. And so we would be talking about that later. So a subclass of immunoglobulin called the IgA or the secretory IgA is being released from the what? From the mucosal membrane towards the outside. And so more amount of antibodies is being formed over here. It's the IgA type of antibody is being formed, and this IgA type of antibody would also interact with the antigen which is being present over here. So that is the mode of action of the mucosal associated lymphoid tissue. So as I told you, you have the M cell. When the foreign particle arrives at the mucosal surface, it contains an M cell, and the M cell will attack which the M cell, it usually lacks the microvilli. And what happens inside this? There are epithelial pockets containing B cells, T cells, and macrophages. And the foreign particle contacts the M cell. It is phagocytosed or endocytosed inside, taken it inside the cell and released into the pocket. And macrophage will engulf the particle and try to degrade it. And an M cell can also endocyte particles and transport them to a cluster of organized cells called the lymphoid follicles. And the B cells in these follicles will react with the foreign particle and mature to form the antibody producing plasma cells. And the plasma cells will leave the follicle and secrete mucous membrane associated antibodies or the secretory antibodies. And the antibody is then transported into the lumen of the gut where it interacts with the particles that causes its production. That is, it will interact with the antigens, the foreign antigens. And so you can see that both intra and interepithelial lymphocytes are being distributed, and they play an important role in the detection of the foreign particles. Now, the next one which we come across is the skin-associated lymphoid tissue. In the skin-associated lymphoid tissue, the skin has having lots of keratinocytes. And so, 90% of the epidermis has been associated with the keratinocytes. And these keratinocytes will produce certain cytokines and cause an inflammatory response when the pathogens, and these cytokines will induce an inflammatory response. During the process of inflammation, macrophages and lymphocytes all will come. So the production of cytokines by the keratinocytes will induce a inflammatory response in the skin. And also within the skin, we also have a type of dendritic cell called the Langerhans cells. And the Langerhans cells, it is a dendritic cell. It is serving as an antigen presenting cell. So what it does is, when an antigen comes from outside, now here you can see the antigen is coming out, the Langerhans cell will internalize the antigen and it will pass, and it will be presented when it moves to the lymph node. You know what's a lymph node? Okay, so it will move to the lymph node and then they will differentiate into the dendritic cell and it will present it to the T helper cells. And the intraepidermal lymphocytes will function as the T cells and activate the B cells to induce an antibody response also. So you have here the antigen presenting cell and antigen presenting cell, dendritic cell, and it is presented towards the T helper cell, and the T helper cell will in turn react with the B cell, and the B cell will also produce an antibody response. So both in malt as well as in the skin associated lymphoid tissue, you can see that the B lymphocytes as well as the T lymphocytes and the antigen presenting cells do play an important role in the presenting of the antigen. So with this, we come to an end of the lymphoid tissues. Thank you for now.